Where does this occur, spermatogenesis, in the male, specifically? Where? No, testes, okay, but be more specific. Where in the testes? The wall of the seminiferous tubules. This process going on in the wall of the seminiferous tubules. That's where the sperm is formed. What occurs in epididymis? Which picks up the sperm from the testes. It's stored there temporarily, and what else? It matures there, okay? So epididymis, temporary sperm storage, and site of sperm cell maturation. But the actual sperm cell formation is in the wall of the seminiferous tubules. That's where this process is occurring, okay? All right, we start, this is all labeled, okay? But obviously, you will see this again without the labels. You'll apply the labels. All right, so we're going to start here with spermatogenesis. That's the process of sperm cell formation. These represent the sperm cells at the end of the pathway. As I said, ignore these. You would not have egg cells in the testes, all right? We start with this cell. This cell is diploid. It's the precursor cell. It's called a spermatogonium. It's indicated by 2N, which means diploid, okay? So this is a precursor cell. It's a body cell, like a typical body cell. It's got pairs of chromosomes, 2N. And again, this is all in the wall of the seminiferous tubule. This cell undergoes mitosis, not meiosis, mitosis to produce daughter cells, okay? One will be a replacement for this. The other is going to be what's called a primary spermatocyte. It too is diploid because mitosis, remember, maintains a chromosome number. To start with a diploid cell, your daughter cells are going to be diploid. Okay? So like I say, this reproduces 2 daughter cells. One will be another one of these, okay, so you can keep this process going. The other one's going to be a primary spermatocyte. All diploid because the division here is by mitosis. You maintain the kind of number of chromosomes. It's this cell, the primary spermatocyte, that undergoes meiosis 1, the first meiotic division. It produces 2 daughter cells, okay? This is the intermediate stage here showing anaphase. Here are the 2 daughter cells. One's already going through meiosis 2, and these are called secondary spermatocytes, and they are haploid. So at the end of meiosis 1, you've got haploid daughter cells. In this case, they're called secondary spermatocytes. And by the way, the number with a degree symbol, number 1 with a degree symbol means primary, number 2 with a degree symbol means secondary. Okay? Now what's happening in meiosis 1? The homologous chromosome pairs, which are lined up side by side, in metaphase simply separate one from the other and go to opposite poles. So you've got these chromosomes, okay, but not only half what you started with. What do we start with up here? Somebody close. How many chromosomes in this cell? It's a diploid cell. Anybody see it? These purple things are chromosomes. Count them. How many you got?
4.
How many do you see there? 1, 2, 3, 4, 5, 6. So 2n equals 6 here. All right? This for a human cell, diploid. How many chromosomes? 2n would equal 46. Okay? That's what I said. All right? All right, we got 6 chromosomes here, and they're paired. Down here, how many chromosomes do we have? How many chromosomes is that cell? Count them. 3. We started with 6, and we reduced the chromosome number by 1.5. These are diploid cells. Now, don't confuse double-stranded chromosomes and single-stranded chromosomes with diploid and haploid. You may have diploid cells with single-stranded chromosomes, or you may have haploid cells with double-stranded chromosomes, like we see here. It's whether or not the chromosomes are in pairs. If they're in pairs, double or single-stranded, diploid. If they're not double or single-stranded, haploid. That's what's going to happen here. Where the cell divides, it's going to replicate its DNA, and they're going to become double-stranded chromosomes, like you see here. Okay? All right. These cells, secondary spermatocytes, undergo meiosis 2. As I point out in lecture, meiosis 2 really involves just simply splitting double-stranded chromosomes into single-stranded chromosomes. You don't change the chromosome number. So the daughter cells from this division, meiosis 2, are called spermatids, and they got the same number of chromosomes as the secondary spermatocytes. The only difference is they're single-stranded now instead of double-stranded. So these two are all haploid cells. Okay? These cells then produce the sperm cells. What kind of division is involved here? Good guesses, mitosis or meiosis? Probably call that a trick question. Look, for each spermatid, you get one sperm cell. No division. It's simply a maturation process. It's called spermiogenesis. Okay? We haven't covered it yet in lecture, but we will today. Okay? So this last step here, going from spermatid, a haploid cell, to the sperm cell, also haploid, it's one for one. No division. It's maturation. And that involves three things. The obvious one is development of the flagellum. So the sperm can be motile. That's one thing that's involved. A second thing, and it doesn't really show well here, it's loss of most of the cytoplasm. You want the sperm cell to be compact. It's easier to move a small cell than a big one. The egg can be big. You want to keep all the cytoplasm, not the sperm cell. It's simply a vehicle to get the male chromosomes into the egg. It's a lot easier to move this small, compact sperm cell, which is not to scale, by the way, if it's tiny. And that's why most of the sperm cell, okay, has very little cytoplasm. So that's the second thing, shedding excess cytoplasm. Form a flagellum, shed excess cytoplasm, and then, unfortunately it doesn't show on the model, development of what's called an acrosome. I'll talk about that in lecture today. Okay? Acrosome is at the head of the sperm here. It's a little bag of digestive enzymes, much like a lysosome. Why? Because the sperm literally has to digest its way through the barriers surrounding the egg cell to get the egg cell cytoplasm to accomplish fertilization. That's why sometimes it needs help from its buddies, more enzyme. Okay? That's all a maturation process. Development of the flagellum, shedding excess cytoplasm to make the sperm cell very compact. And again, this is not to scale, unfortunately. And then form that acrosome, little bag of digestive enzymes. Okay? And we'll talk about this in lecture also.